Okay, we are live. All right, so it's 6 o'clock in the UK. I'm Tom Ellis, and I'm introducing today's speaker, Ron Weiss, for SynBIC and SynBioUK seminar. We can probably wait around for a minute or two just as attendees arrive. I think, from what I can see here, there's we're getting well into a good number of attendees, and let's see, yeah, more keep cropping up. So let's just hold on a couple of seconds, and then we can get going. All right, so I think a lot of people are going to be very excited to hear the science today from Ron. So let's get going as quick as possible. So this SynBioUK SynBIC seminar has our guest speaker, Ron Weiss from MIT. He's the director of the Synthetic Biology Center at MIT. I have followed Ron's research for many years. He's one of the real pioneers of synthetic biology, coming from an electrical engineering background. I believe, if I'm not wrong here, that Ron did his PhD at MIT with Tom Knight in an electrical engineering setting and converted an electrical engineering lab effectively into a wet lab that, among other things, led to the invention of the BioBrick, Tom Knight's group, and the instigations of iGEM. I think Ron was doing SynBio in the 90s, which is pretty rare. So maybe like 1997, 98, I think. If I recall correctly, something SPICE, was it a computer program language?
Yeah, I had BioSpice, yep.
BioSpice, yeah.
Programming language for bacterial colonies. That
Yeah.
was the last chapter of my thesis.
Very cool. Very, very ahead of its game, right? Ahead of its time. And so Ron was then a member of faculty at Princeton for many years. And I think at the time when I was a postdoc in Boston, everyone in Boston was very excited when Ron came, and I think similar time to when Chris Voigt came as well, over to MIT to begin the Synthetic Biology Center at MIT, where Ron has really pushed the limits of what synthetic biology does in mammalian cells, looking at pattern formation, logic, and particularly differentiation using synthetic circuits in mammalian cells. And Ron, it's a shame we can't have you here in London. I think last time you came,
yeah,
you gave a great talk, and I think you even sneaked off to Bristol and went surfing on the day before your talk or something.
Yeah.
You might have had a good day of that today, because we've had some really high winds here, so the seas might have been quite good if you'd been able to come.
I would have stayed more day. Well, it's an outdoor, it's a wave pool, so it's not actually.
It's artificial.
It's an artificial wave. It's pretty amazing. I recommend everybody try it out.
I bet it's probably a lot cleaner than the Irish Sea. So.
Yeah. Very consistent.
Yeah. Okay, so Ron, if you're ready, Ron's going to talk about programmable organoids and cancer immunotherapy and his group's research in this area. There's going to be some time for questions at the end. Please write your questions in the Q&A, and at the end I'll be moderating, reading out those questions and getting Ron's answer to them. Ron, it's over to you now. Your slides are up, so take it away.
Great, thank you. Good to see you again, Tom. And it was really fun to come visit you guys slightly before the pandemic, and then we talked about coming back to visit again, not just because of Bristol. I wanted to hang out with you guys too. And yeah, so we'll do it via Teams or this time, but hopefully there'll be another chance to come and visit later on. And what I'll do today is, for the most part, I'll try to present a lot of new slides. I think the majority of my slides today are new from last time, and I'll also try to keep my introduction fairly brief, so we can dive into the science. But I do have to give kind of my, spiel about, the fact that I view synthetic biology as being the ability to program cells, which is something that's shared among many people in synthetic biology. So it's really exciting to see the progress since the very early days now, a quarter century ago, when I started, 1996, is when I started working with Tom Knight on genetically programming cells and thinking about programming abstractions for bacterial systems. And it's really been amazing to see, all the progress in the field, and all the young people entering that. And so the notion of coming up with a high-level language, being able to create some kind of a regulatory network that is able to implement that high-level behavior, putting that on a piece of DNA, or now RNAs become a really interesting medium for programming cells, and being able to embed that in cells as a way of extending or modifying the cells. And I would say when I started out as a computer scientist in synthetic biology, I was hoping to kind of find this, or be able to create this kind of modular, approach where we can, plug and play and replace things and write fairly complex functions. But basically, as I've gotten older, more gray hair, more backaches and so on, I've basically come to appreciate and respect biology a lot more. And so this notion of, the fact that the biological systems are really incredibly complex, right? And, this argues for, first of all, an important appreciation, and also the fact that we may not be able to just do anything that we want to them too easily. But at the same time, this also argues for synthetic biology, because synthetic biology does provide very sophisticated mechanisms to be able to address and possibly even come to match the complexity of the underlying biological system. And so that's a premise that I'm saying is not that the complexity of underlying biology actually is an important argument for using synthetic biology, because synthetic biology has the ability to implement sophistication for controlling and addressing developmental pathways and other kinds of disease states in a way that I would argue, not any, possibly no other technology provides. And so hopefully I'm preaching to the choir at this point. But one of the things that we have begun to do more recently is begin to think about, what is a high-level abstraction, like a simple high-level abstraction that we should use every time we begin a new project in synthetic biology. And that's basically the 5 W's and 2 H's. And this is something that we try to answer when we initiate any new project. So the notion of asking the question, who are we trying to engineer or affect? What kind of cells? What is it that we're going to be expressing? When are we going to express particular genes? For example, in an organoid, when are we going to express particular genes that drive various stages of the developmental pathways? Where are we going to do this? The spatial aspects of the system are really important. Which part, let's say, of the organoid? Why are we going to do this project, for example, with programmable organoids? So you can argue, this is for disease modeling. This could be for drug development. This could be for helping regenerative medicine. But on a more technical level would be, why are we expressing gene X? And that could be based on a variety of intracellular biomarker levels. So there could be a very technical reason for that. How are we going to do this? For example, which particular, are we going to use transcription, translation, other kinds of regulatory mechanisms, and how much? So, synthetic biology, contrary to what maybe people from the outside think, is not just a digital approach, right? So oftentimes we think about kind of more analog aspects of synthetic biology and not just kind of on-off behaviors. So these questions really frame a lot of the things that we think about when we begin to consider new projects, and I'll use that a few times along the way today. And so, what's been now accepted essentially in the central dogma in synthetic biology, the notion of having sensing, processing, and actuation mechanisms, is now allowing us to basically address these fairly complex questions. And so, we oftentimes think about inducible gene expression, multi-input logic, toggle switches, oscillators, and feedback regulation, and those really comprise the basic tools in synthetic biology. But today, towards the end of the talk, I'll also add the notion of perceptrons or building blocks for neuromorphic computing, arguing that this is also going to be an important building block moving forward in synthetic biology. And so the notion is that we can define a high-level behavior, convert that to a regulatory network. And again, we always have to remember that this regulatory network is going to exist in a very complex environment that we only understand to a limited extent. And so the other perspective on synthetic biology is this notion of bottom-up, that we start with DNA assembly, we build regulatory mechanisms, transcription, translation, post-translational control, allowing us to build the basic modules. To me, the thing that is super exciting about synthetic biology is the ability to really program. Okay, so go beyond the individual modules to things that really begin to constitute quote unquote real programs with sophisticated behavior. And so I'm going to talk about our ability to finally be able to create multi-step programs for programmable organoids. And that's something we've been very excited about over the last several months, or a little bit more than that. We do work quite a bit on feedback control. I won't talk about that too much today. And then the other one is adaptive systems. So can we create high-level systems that can adapt and maybe improve themselves? And so we're beginning to take some initial steps towards that with the neuromorphic computing, which will be the last vignette that I'll discuss. And so the point is that these sophisticated programs now are allowing us to address a variety of application areas in ways that were not possible before. And I'll focus mostly on regenerative medicine and organoids. I'll touch very, very briefly on cancer immunotherapy, but we also work on vaccination and other kinds of indications. And so the three vignettes that I'm going to discuss today: programmable organoids. I'm going to talk about some work that we're doing with respect to building fast phosphorylation circuits, and then finish by talking about the neuromorphic computing. And as I mentioned, a lot of what I'm discussing, you should keep in mind the answers, or posing the questions of the 5 W's and 2 H's. So for example, where are we going to express genes? Maybe in a cell lineage specific way for organoids or for cancer cells. What kind of genes are we going to express? So for organoids, it's going to be cell-fate regulators. How are we going to do this? I'll actually demonstrate both, not just transcriptional control, but also translational phosphorylation and DNA recombination as mechanisms that we're using as building blocks. And as I mentioned, not just how much, not just on and off, but why graded expression may be quite important. And finally, I'll also address responses that are either in the hours, or with a case of phosphorylation circuits, it's for a second. So I'll start off with talking about the programmable organoids. And so actually going back, all the way back to my PhD about a quarter century ago, when I got started in synthetic biology, I was looking at ways of actually the opposite, which I was looking at ways by which biology can inspire me to think about how to program computers. And so I was looking at embryogenesis as a really interesting example where you have, hundreds of thousands, millions, hundreds of millions of elements that really interact together and create very complex, robust behavior. And so that was super exciting to me, and I was creating a variety of different simulations to model embryogenesis. And at that point, I actually decided to flip the arrow, and rather than take what I know or get inspired by biology to program computers, I wanted to do the opposite, which is take what I know about computing and try to program biology. And at that moment, I said, wouldn't it be really cool if we can, for example, program tissues by design? And so that's something, to one extent or the other, I've been interested in, for really quite a long time. And so the notion here, ultimately, is that we'd like to take embryonic stem cells, or now induced pluripotent stem cells, genetically reprogram them, or genetically program them to create all kinds of patterns, could be 2D patterns, could be 3D patterns, and then use those patterns as a mechanism to create, for example, organs on a chip for drug development, or create organs or organoids that we would be able to actually use for engraftment, to actually implant these to aid in regenerative medicine. And so the question here is, can we create genetic programs that actually drive and guide this entire process by having spatial, temporal, and cell type specific control? And another argument here, why is it that we need synthetic biology? Because we're actually trying to recapitulate embryogenesis in unnatural environments. So if we're trying to grow an organoid on a chip, that's really not the environment that these stem cells kind of know how to create tissues or organs. And so they may not even have the capacity to be able to do that, even if we provide cues from the outside. And so the argument here is that we really have to rewire these cells. We have to provide additional instructions so that they're able to develop properly in these unnatural environments, and also to be able to support co-development of all the appropriate cell types. So we can't really control all the individual cells from the outside. We need to embed rules in them so that these cells can co-develop. So all the cells that are necessary for the development of a particular organ actually have the ability to interact with each other and create the final structures. And so I'll talk today about one of our target organoids, which is a liver. And as we've gotten, as we've done work in this liver, really appreciate more and more the complexity of this organ. And this is, quite an important organ to be able to make, both for drug development. Obviously for regenerative medicine, there's a huge need for liver transplantation, and for drug development, this is really a critical organoid due to the fact that many of the drugs that we take end up in the liver. It has the function of being able to take blood from various organs, gastrointestinal tract, purify that, and then basically provide that back to the heart that's going to oxygenate it. And so, looking at it, it has some really complex architecture. It has vasculature that is really throughout the entire organoid, and that's essential for its purification, blood purification purposes. And it has these liver lobules that have some really interesting hexagonal structures that provide a mechanism for the blood, the dirty blood, to come in, get purified, have the waste basically get secreted through these bile ducts to the gallbladder, and then ultimately as waste product, and then come back to the heart. And so, we'd like to be able to recapitulate some aspects of that as much as possible in vitro and ultimately in vivo. And need to also remember that there's the blood itself is really complex, comprising many different kinds of immune cells. So that's such as the Kupffer cells, these macrophages that really play an important role in many liver diseases. So our ultimate goal is to be able to recreate this kind of structure. And so synthetic biology, again, arguably can provide the notion of really powerful synthetic developmental pathways, and can be used also to understand in drug development, what happens in real time as we apply particular drug candidates. It can tell us, for example, the hepatocytes are reacting in a particular way by having these genetically encoded real-time live cell sensors. And ultimately for organ transplantation, we'd like to be able to create a liver that can better engraft and maybe even have some improved metabolic function. And so we started really focusing on synthetic developmental programs about 15, 16 years ago, after we were able to create a variety of different patterns in bacterial systems. And we said, what would be fun, what would be useful to be able to do with stem cells, now that we've been able to have some kind of control over bacterial systems. And we thought that would be interesting to be able to create a self-type multi-step beta cell differentiation program, where the notion is that we would try to recapitulate the important steps as cells in normal embryogenesis develop from the inner cell mass, from the stem cells, along and support all the important stages along the way: endoderm, pancreatic progenitors, endocrine, and beta cells as a way to hopefully, recapitulate proper development of insulin-producing pancreatic beta cells. And so we imagine having this multi-step program where we initiate the first step to endoderm, and then from endoderm there will be an automated step where cells would notice that they, detect that they're endoderm, drive towards pancreatic lineage. When they get to pancreatic progenitor lineage, they would drive themselves towards endocrine, and so on and so on. And so basically, the cells would take the next action when they were ready to take it. And so, this is a project we started about 15 years ago, and then finally, we now have the ability to actually do automated multi-step differentiation. So I'll discuss that in a few slides. And so it started with work by Patrick Gee, and we ended up collaborating also with Linda Griffith and their former postdoc Bo. And the first question was, can we go from induced pluripotency stem cells to endoderm genetically? And this is not something that was known. And so we tried a variety of cell-fate regulators, including GATA6, and it turned out the GATA6 was the one that actually worked. And so what we did is we engineered GATA6 expression in a population of iPSCs. We're using lentivirus, and what happened was that lentivirus integrated a different copy number across the population of iPSCs. We said, well, this is not ideal. We typically like to have in synthetic biology very tight control, but we said, let's go ahead and see what actually happens with this heterogeneous iPSC population. They're actually expressing GATA6 at different levels across the population. We said, perhaps not ideal, but this is what we have. Turns out that diversity of GATA6 expression levels was really critical for the success of this project. And so day 0, there's not much going on, but by day 5, we see this really nice clustering and symmetry breaking that's taking place. And we analyze this further, and we realize that we're actually able to generate endoderm, mesoderm, and ectoderm by having control over the heterogeneity in GATA6 expression level. And so that allowed us to actually go for step 1 in this process from iPSCs to these germ layers. We wanted just endoderm. We ended up getting more than what we bargained for, but we had a very controlled way of doing that. At that point, I told Patrick, okay, cool, we got step 1 to work. Let's go ahead and work on step 2. So luckily, Patrick didn't listen to me, and he was actually really curious to find out what happens after you push the system towards step 1. What happens? What happens when biology takes over? And it turns out that there's actually some default programs that are taking place here, and the endoderm continues to develop towards hepatic lineages and creates actually hepatocytes and cholangiocytes. The mesoderm ends up creating hemangioblasts, but actually ultimately vasculature and hematopoietic progenitors. And I'm not going to talk too much about the ectoderm. That's more in the 2D environment, but a little bit less in the 3D environment. And so we ended up actually getting a liver organoid, vascularized liver organoid that contains all the cell types that are known to exist in a fetal liver. And so we're actually curious, so now we have all the major liver cell types. What about the architecture? So these liver organoids, they're vascularized, and they can grow really large. They got the hepatocytes have the right morphology to them, and we're seeing immune cells such as Kupffer cells, and actually maybe some initial bile duct formation. So we're really excited about that. And so besides cell types architecture, we also measured liver function, and albumin and urea levels were really quite nice. They're really at primary hepatocyte level. So this is, in fact, an organoid that industry, big pharma is actually interested in using right now for their drug development purposes. As we analyze this organoid further, we realize that it has some adult properties, but also some fetal properties. So in particular, cytochrome P450, which is an important liver enzyme, its levels were really lacking. So even though this is a really nice developing organoid, it's not a perfect adult liver, not surprisingly. And so we asked the question, can we engineer multi-step differentiation that now would allow us to create automatic maturation of these of the liver, and in particular hepatocytes. So we wanted to be able to create a mechanism, a biological program. I showed you step 1, that would be able to carry out step 2, which is basically to detect that if a specific cell has become a hepatocyte, express hepatocyte maturation transcription factors, and make sure that these factors are expressed only in the hepatocytes and not in any other cell type in this developing organoid, because those would be detrimental to the other cell types. So we wanted to first of all have the question, can we detect that a cell is a hepatocyte versus other cells? So we discovered that microRNA 122 is a good indicator for that. And then we identified a variety of different hepatocyte maturation transcription factors. And we've had a lot of experience with microRNA sensing, and we said, okay, hopefully this won't be such a big deal to be able to express hepatocyte maturation factors only in the hepatocyte. This is something that we actually struggled with for years, and so it wasn't quite working. So here's a depiction of a microRNA sensor. This is a low sensor, so in this particular case, if the microRNA is low, it targets the messenger RNA. In this case, would be an MK protein. So high microRNA will result in low MK expression and vice versa. So we don't want a low sensor. We actually want a high sensor. And we've created high sensors like this, where we would have a microRNA, for example, microRNA 122 would repress a transcriptional repressor of the final gene of interest, such that microRNA binds the messenger RNA, and then we no longer have repressor, allowing MK production to go up. And we actually tried this, and the problem is that this did not work. Okay, what turns out to be a problem is epigenetics. So epigenetics is basically the silent killer in a lot of mammalian synthetic biology. And so this is not something that we just ran into. This is another paper that looked at hepatocyte differentiation from human pluripotent stem cells, and they realized that their genetic circuits were being silenced. So it was not possible to create something that would allow activation at future stages of the differentiation. And so they were stuck. We were stuck with this for a while. And what happened is we actually ended up relying on another project that was taking place in the lab. And specifically, this is a project by Bree, a former graduate student in the lab, and Noreen, who is a current graduate student. And so they were tackling this problem of epigenetic silencing within a different context. So really interested in cell-based therapies and biomanufacturing, where epigenetic silencing also is highly damaging to our ability to regulate things, to regulate expression. And so the point is that once we downregulate transcriptionally the expression of a particular gene, expression goes down, but we have a really hard time and sometimes completely unable to turn it back on due to the epigenetic silencing. And this is another paper showing that epigenetic silencing is a big problem. The longer you turn gene expression off, the more difficult or impossible it is to turn gene expression back on. And so we decided, rather than to rely on transcriptional regulation, let's continue to express our genes constitutively, because if you express them constitutively, then they don't tend to get silenced, but rather do control at the translational level. So regulate things at the RNA level. And so this is my requisite CRISPR slide. So any synthetic biology talk, I think by law, has to mention CRISPR. So there you go. And so here we're actually not using Cas9 or dCas9. We're not trying to modify DNA, but rather we're looking at regulation at the RNA level. So these are CRISPR enzymes that process RNA. And what we're doing then is we're using these CRISPR enzymes to be able to downregulate at the translational level. So the notion is we have a reporter, and we have shown that for this library of CRISPR enzymes, these endoribonucleases, when you add the endoribonuclease, you see really nice knockdown in gene expression. So we're getting between 40 and 250-fold repression in these mammalian systems, which is really quite nice. So we have an off switch mechanism. using endoribonucleases that would work with constitutive expression. But we also were able to create an on switch. So the way this on switch works is that we put RNA degradation signals on the transcript. And so normally when we have expression, the RNA is degraded, so the expression of the reporter is low. However, when the endoribonuclease arrives, it binds this synthetic hairpin, which is the recognition sequence for this endoribonuclease. It cuts off the degradation signals, and then we have another RNA element called the triplex stabilizer that stabilizes the messenger RNA, such that when we cleave and remove the degradation signal, we actually end up getting high level of reporter expression. And so in terms of the experimental results, we start off with low levels of expression, and then we add the endoribonuclease, and we see that we have a nice increase in expression across the different constructs. So sometimes it's as low as 3-fold, but we're able to get up to 200-fold activation with this endoribonuclease on switch. So they're also orthogonal. So this PERSIST platform provides for subsets of endoribonucleases that really have nice orthogonality. You obviously have to be careful about which specific subsets you're using. Some of them do have crosstalk, but for many purposes, you can find appropriate sets of endoribonucleases that have nice on-off behavior and no crosstalk behavior. And we've also been expanding this library of 9 different ERNs. We have about 5 or 6 additional ones that are showing, pretty nice behavior. So they're orthogonal. And so now the point is that rather than using transcriptional repressor, we're actually using constitutive expression of our elements, this Cas-E endoribonuclease, which results in downregulation of the transcript that's coming to express the gene of interest. So the microRNA knocks down Cas-E, which allows MCAT expression to actually take place. And so we indeed implemented this in our organoids. Previously, we would have these genetic circuits in the organoids that would get silenced after about 3 or 4 days, and they just were not useful for a multi-step differentiation. Now that we switched over to constitutive expression and translational regulation, we are able to have circuits that operate for many weeks and possibly even months. And so in particular, we have a mechanism, a circuit, where if microRNA 122, as I mentioned, the hepatocyte microRNA is low, then we're expressing a puromycin. We're also expressing a blue fluorescent protein, which helps us do the initial selection of integration of these elements. And now, when microRNA 122 is high, it downregulates Cas-E, and that allows expression of the cytochrome P450 gene that we were missing badly, as well as some additional genes, PROX1 and ATF5, that are also useful hepatocyte maturation genes. And indeed, we're able to see nice expression. We also are fusing cytochrome P450, the CYP3A4 gene, with an MKO2. So we see nice expression. This is day 12, but we see good expression beyond day 12, which I'll show you in a second. Urea production goes up, which is okay. Our urea production was already quite good. It goes up even a little bit higher. Albumin production goes up slightly, which is great. And then the CYP3A4, you see that CYP3A4 function in terms of measured luminescence goes up, really quite nicely, 5-fold. This is about, day 15 or so. And then as we look further and further, you see that by day 12, we have a 10-fold increase of CYP3A4 expression levels due to this microRNA 122 sensor, and by day 25, we have a 100-fold increase in CYP3A4 levels. So this mechanism seems to be able to combat and resist epigenetic silencing really quite nicely. We have very robust gene expression. So we are very excited about now the ability to carry out multi-step differentiation, and we want to use it all over the liver organoid, as well as begin to apply it to other kinds of organoids as well. And this PERSIST platform that we're using here allows us to do even more complex logic. So here, for example, I'm showing you all the 16 two-input logic functions. In particular, on the right side, I'm showing you the B and not A function. So B removes the RNA degradation, and A is there to cleave at the 5'. And so this is combining an on switch and an off switch. And so the only time we're going to see expression here is when B is high and A is low. So this is one example. We can do an OR gate by having the recognition endoribonuclease recognition sequences right before the degradation signal, and we can have a not OR gate by putting those recognition sequences at the 5', such that if either A or B are present, that is enough to knock down YFP expression. So we have logic functions that are supported by these endoribonucleases. We can compose them. So here we're showing a cascade of a not or repressor two-stage cascade, and this works nicely. So this is a negative control where we are not connecting the input to the output, so it remains high. But now when we connect the input to the output, we see the desired basically rise in output expression as we increase the amount of input in the system. And we can modulate that by having different variations on this two-stage cascade. We can get different kinds of responses in the system. We also can have an activation repression cascade that works nicely, negative control, and now as we add the intermediate element, we see that basically as we have more of the input, we have less of the output as expected, and in the same way as above, we're able to modulate the dosage response of the system quite nicely. And we have really nice dosage response, 100-fold on-off cascade response in the system. So basically, we are able to compose them with high performance. And the other thing that's interesting about this PERSIST platform is that one endoribonuclease can function both as a repressor and as an activator within the same circuit. So here we're showing you an implementation of a toggle switch, a bistable toggle switch, whereby each element activates itself and cross-represses the other component, and that allows us to create a nice, compact, and robust system that has two steady states. So if we start off with not providing any input to the system, we see that the system bifurcates, that a portion of the cells become high for one state, a portion of the cells become high for the other state, and essentially very few cells have expression of both. And this is without putting any input at all to the system. And we're able to modulate the switching by being able to select different different ratios between the two different components, the two endoribonucleases CSY4 and CASE, such that we can get the initial fraction of the cells to be either mostly yellow or mostly red by being able to modulate the expression levels. Let me now switch over to the second vignette, which is phosphorylation. I'll go over this quickly. So I want to be able to talk about both the phosphorylation circuit and our recent work with neuromorphic system. So here, I'm going to show you phosphorylation circuits that are able to respond in seconds. And the reason we want to be able to build phosphorylation networks is to be able to modulate or modify existing pathways that allows us to couple circuits into important cellular signaling pathways. Certainly, we would like to build to understand, and there was an element of this project that I'm not going to get into due to time, that the construction of the circuits that I'm going to be discussing really helped us gain new insight about endogenous signaling pathways. So I won't talk about that today, but it was really rather interesting. And also the fact that these are fast. The fact that these can actually switch, can have an input of 1 second and actually switch their behavior. And this is not something that's just simply not possible with transcriptional control or translational control. And so while there have been, lots of demonstrations of engineering protein-protein interactions, there hasn't been any demonstration of circuits that are purely based on reversible protein-protein interactions. So Michael Elowitz had this really elegant CHOMP system that is based on protein cleavage. We also, with Velia, that many of you know, she spent time in Imperial, also had a system based on this protein cleavage, but these are irreversible and hence are not able to be fast. We wanted to focus on reversible fast protein-protein interactions and create a full circuit that's purely based on them. And so to do that, Deepak, who's a postdoc in the lab, implemented a system that we're calling phosphorylation-required interaction and mediated effect, PRIME, where the notion is that we will have an input signal that phosphorylates a binding domain, which causes the upstream and the downstream elements to come together. These are all, these are things these are chimeric proteins that we put together, and as a result of this phosphorylation-required binding, the upstream effector is able to dephosphorylate or phosphorylate the downstream effector, and hence we have a phosphorylation-dependent signal propagation, either repression or activation. And so to do this, to build circuits, we need building blocks. And one of these building blocks, an important building block, is an OR gate. And so the way this phosphorylation-based OR gate works is that we have two elements that have, in this particular case, a phosphorylation to a histidine kinase response regulator, and both of them are then able to phosphorylate the downstream element. So if either one of these upstream elements is phosphorylated, that is enough to phosphorylate the downstream element, and that allows us to create an OR function. To test this OR function, we needed to build actually a rather complex circuit, almost as complex as some of the later circuits that I'm going to be showing you. In this particular circuit, either the input IP, this is an extracellular ligand, is high, or small molecules DOX and galactose have to be high. So it's basically implementing IP or DOX and galactose. And I won't get into the entire circuit and how it's built. I will just show you the experimental results whereby if that is not satisfied, the logic function is not satisfied, the output is low. But if either IP or DOX and galactose are high, that's enough to drive the downstream phosphorylation process, which we're then able to detect. In this particular case, we're connecting it to gene expression, and we're able to detect high levels, and that is true for all other cases where IP or DOX and galactose are high. The other element, the other device that we built is a NOT gate. And so the way this NOT gate works is that if the upstream element is not phosphorylated, that allows the downstream element, which is this HOT1 JH1 chimeric protein, to be autophosphorylated. Okay, so low input results in high phosphorylation output, but when the input is phosphorylated, this HOG1 chimeric protein translocates into the nucleus, binds HOT1, and this phosphatase domain basically dephosphorylates JH1, and we have dephosphorylation of the output. So when the input is high, the output is low. We have a dephosphorylation of the output. This is the circuit that we had to build to be able to test this out. And the logic here is that if DOX and galactose are high and sorbitol is not high, then the output can be high. And indeed, when DOX and galactose are not high, the output cannot be high, as I'm showing you with all of these conditions. And when DOX and galactose are both high, then the output depends on the presence or absence of sorbitol that you're seeing nicely here with the experimental results. So now we have an OR gate, we have a NOT gate. What can we do with these systems? And we said, let's go ahead and try to build a circuit that's fully based on phosphorylation interactions. And we said, let's go ahead and build, a toggle network. And so in particular, the high-level design is that we have two inputs, IP and sorbitol. These are the small molecules that I've described in the previous set of slides. And then we have this cross-repression topology where YPD1 ultimately represses PBS2, and PBS2 ultimately represses YPD1. And the inputs IP and sorbitol basically repress one branch or the other and allow us to flip between the two toggle states, such that either YPD1 is high and remains high unless sorbitol is provided, or PBS2 is high, remains high until IP is provided. And so this is the high-level mapping into basically a not-or network. We do not have not-or implementations. We have not and ors. And so, in fact, we had to implement it using ors, nots, and also buffer gates between them to be able to connect all of these phosphorylation, dephosphorylation mechanisms. And so the ultimate circuit, genetic circuit that's based purely on phosphorylation looks like this. So a lot of interactions here, a lot of phosphorylation, dephosphorylation interactions, also detection of inputs from the outside. So this is quite a large circuit. It might be the biggest circuit that's out there as far as I know, but at a high level, this circuit is actually not overly complex. We have the YPD1 branch, we have the PBS2 branch. Only one of them is going to be high, and it's going to dephosphorylate the other one and maintain its high state. And so high state can be either YPD1 high or PBS2 high, and that will maintain state hopefully forever. And when we introduce sorbitol to the system and then take it away, or IP into the system and then take it away, the circuit is able to maintain its behavior. And the way we can visualize that is by looking at a HOG1 that is fused to a GFP. This is actually, by the way, this is done in yeast, and we can see that nuclear phosphorylation of HOG1. We can see the ability to have a very short sorbitol impulse to get the high state, a short impulse of IP to get the low state, and on and on. So we can basically turn this on and off, essentially as many times as we want. It is ultra sensitive. I think for the sake of time, I'm going to not discuss this in detail. I want to talk about the neuromorphic circuits, but it is able to detect very low concentrations of sorbitol and very short impulses. Here I'm showing 2 seconds. We can go down to 1 second. We can probably even go lower than 1 second and still have the ability to permanently flip the toggle switch. Okay, and so I'll skip over the explanation here. And so the variety of different applications that we're interested in that would benefit from the speed of this protein phosphorylation type of circuitry. Whether it's for differentiation possibly, but for fast sense and respond where you can't wait minutes, hours or minutes. You have to respond within seconds. There could be many different situations, therapeutic situations where that's the case. So let me now shift to the final vignette that I'll discuss briefly, where the notion is to be able to create mechanisms that control gene expression, but not to do it using the abstraction of digital logic, but rather to be able to use the abstraction of artificial neuronal networks inside cells. Okay, and so no need to emphasize that artificial neuronal networks are everywhere. And by the way, this is a project done by Andrew, former graduate student in the lab, Christian, a postdoc, Wenlong, a postdoc in the lab, and also in collaboration with Elisa Franco over at UCLA. And so we got excited about basically the fact that artificial neuronal networks have their motivation in biology. Let's bring it back to mammalian cells. And importantly, the focus here is not to recreate digital logic, but really try to focus on what are artificial neuronal networks really good at. And they're really good at pattern recognition. And so that's what we wanted to focus on, is how can we do pattern recognition circuitry using this abstraction. Why do we want to use artificial neuronal networks? A variety of good reasons for that. We would argue So digital and analog circuits are, are hard to build. We know this. They require all kinds of different kinds of, basic building blocks. Whereas artificial neuronal networks, they have the same repeated node over and over and over again. This notion of a perceptron, which I'm going to talk about in detail. So the architecture in principle is simple. It's all about the connections. You can create compact implementations where similar logic functions would require really huge circuits. Oftentimes, it's in digital logic. There's a lot of effort that goes into composing gates into complex function. So we have to spend a lot of time. This is actually going back to my PhD. Half of my PhD focused on modifying the input-output responses of an OR gate, of a NOT gate, to be able to get it so that we compose these NOT gates into functions. And this is showing some really cool work by Chris Voigt to be able to do that. But it's still hard to be able to generate gates that are matched properly to create the desired large-scale logic functions. Whereas with artificial neural networks, what we do is we can optimize the weights of the connections in a rational way. And this weight optimization is also very cool because you can take a given topology and you can modify the weights and create a very different kind of computation, which is something I'm going to show you with respect to basically doing one type of pattern recognition, modifying weights and topology, and getting a very different kind of pattern recognition. And so these artificial neuronal networks are based on this repeated node, this perceptron node, which does, computationally, a fairly simple function, which is doing a weighted summation of the inputs, where the weights can be positive or negative. And so you can have addition and you can have subtraction, and then this thing goes through what's called an activation function to give you the final result. So basically, can we implement weighted summation with positive and negative weights? Now, I mentioned this activation function, so you want to then take this weighted sum and put it through an activation function, which basically reshapes that result. So in situations, for example, where you get a negative value, that's rather difficult to get in biology, to get negative concentrations. And so what actually turns out to be a very useful activation function in artificial neuronal networks, and so this was not, looking at this because of biology, is this rectified linear unit. So this notion that you can have a weighted sum whose value is negative, in which case it gets mapped to a 0, and then otherwise if it's positive, then it just gets mapped, with a linear function. Okay, so this is called a rectified linear unit. And the nice thing about this rectified linear unit, or ReLU, this is something we can actually implement in biology, which I'm going to show you. And I'm also going to show you these sigmoidal activation function. And so these ReLU functions, if you can actually implement them in these neuronal networks, perceptron networks, are really good because they allow you to train the network and get it to recognize different kinds of patterns or do different kinds of computation. So this is a really important building block. So let's now talk about pattern classification. So this is really one of the more important things that we do in synthetic biology. So the notion of taking a system that has particular biomarker levels, for example, we would say we only want to have output when the combined biomarkers are low or the combined biomarkers are high. And so we'll call this a dual region. Or alternatively, we want to create a situation where we want to only express the output when the combined biomarker levels are intermediate. So we'll call this a bandpass. And so neuronal networks provide a really powerful way to do this type of classification to be able to recognize specific patterns. So here I'm showing you an abstraction of a neuronal network. This is a 3-node network. It has 2 inputs, x1 and x2. And the notion is that x1 and x2 go into node 1, they go into node 2, and then node 3 combines these 2 nodes, these 2 computations. And so with this topology of 2 inputs, 3 nodes, what kind of patterns can we recognize? And so the way this thing works, the way these pattern recognizers work is really, really interesting and powerful. And as you spend more time with neuronal networks, you get to appreciate it more. So here in this case, let's say node 1, we engineer that node to be able to, it does the function whereby as you have more x1 and x2, you have less output. So x1 and x2 would have negative weights. So you start with a high value, and with these minus x1 minus x2, that's the final output that you get. So you get this, what's called bias. So b minus x1 minus x2. So that would be our node 1 computation. Node 2 computation would be start off low, and then the output is x1 plus x2, but also subtract this bias. And if the level is below 0, then the output is 0. Okay, so this is giving us an ability to have 2 different pattern recognizers or pattern classifiers. And so now if we wanted to detect this dual region, what we can do is we can basically add up the recognition by node 1 with the recognition by node 2 and subtract this bias. So that's what you're seeing here. N3 equals N1 plus N2 minus this bias. And then if it goes lower than 0, then this is just a 0. So this is a dual region recognition mechanism. We can take the same node 1 and node 2, and by modifying the way this node 3 operates, we're going to say, and the weights of node 1 and node 2 going into node 3, we can basically do the computation B2, the initial bias, minus N1 minus N2, and get the bandpass recognition sequence. So we're able to do, let me show you a couple of slides. I think I'm unfortunately running out of time a little bit here. So we can do a weighted addition. So weighted addition is simply when we have 2 DNA inputs, and we have expression, which is the final reported expression is simply the summation of those 2 inputs. And so this, as input 1 and input 2 go up, the final input goes up as well. So this is fairly simple. But then what about negative weights? How can we do subtraction? And so we can take this endoderm mechanism, and we can actually use that to do subtraction. So in this particular case, we're going to have x1, x2, x3 as the inputs. So we have x1 plus x2 minus x3. And so the way we achieve the minus x3 is by having the endoderm sequester away this RNA. This is a single turnover enzyme, so it's not catalytic. So it's actually going to act in the desired fashion, which is acting as a subtractor. And so we get this nice ReLU function from the system where here I'm showing you, x1 plus x2 minus x3, and we can see that it has the desired ReLU function. We also did a sigmoidal type of activation function, which is based on recombinase sequestration. This is something that unfortunately I'm not going to have the time to discuss, but not only can we achieve ReLU activation function, we can also achieve sigmoidal activation function. And then we can actually realize this by this. I'm showing you here again the endoribonuclease. We can measure this endoribonuclease activity, and we see that it has the desired response. The more reporter we have, the more expression, but then the more endoribonuclease we have, the less expression we get. So this gives us the desired response experimentally, and indeed we are seeing the ReLU-like function. And similarly with the recombinase, I'm not going to get into now, we are able to show this weighted addition and weighted subtraction, and now with sigmoidal behavior. And then we're able to fine-tune, so I'm running out of time, we're able to fine-tune experimentally the dosage response of these systems in a very controlled way. So we can fine-tune the decision boundaries by modifying various weights in the system. And we are also then able to create multi-input classifiers. So this is a 2-input cancer cell classifier that can detect the levels of microRNAs and only have a high output when the 2 microRNAs are high. So again, I'm not going to have time to discuss that. And we're able experimentally to create multi-node artificial neuronal networks using these sequestration mechanisms inside individual cells. And so this is simulation for HEK cells, but also experimental results in HEK cells showing that we can have this type of bandpass detection based on the mechanism that I described in the very beginning, as well as a 3-node artificial neuronal network based on sequestration that is able to do experimentally this dual region recognition. Okay, so we're able to show the ability to have single perceptrons that we can modulate their decision boundaries, and then we can have multi-input perceptrons for microRNAs and have 2- and 3-node perceptron networks that operate inside individual cells. So quick summary, I showed you that for programmable organoids, gene expression heterogeneity is a very useful building block, not just on and off. Our PERSIST platform is able to overcome epigenetic silencing. I showed you fast phosphorylation as a way to build circuits, and then artificial neuronal networks as, I believe, a new frontier for synthetic biology. I talked about the people that have done the work. I mentioned them throughout. I want to thank you for your time, and also mention that we have several positions now available in the lab in foundational synthetic biology, in programmable organoids, and in immunology. So if you're interested in this mammalian synthetic biology efforts, please do send me an email. So thank you for your time.
Awesome. Really great talk, Ron. Thanks for giving us that talk. I was enjoying it's evening here, so I was enjoying it with a beer, and I don't know if you see him behind me, but Giebart Stan was here as well, having a socially distanced beer with me whilst watching yours through the glass window, so all safe. Okay. Really amazing to see this unpublished stuff, particularly the neural network work, and the PERSIST toolkit was fantastic. There were lots of questions appeared from the SynBioUK and SynBIC students, and I'm going to read out the ones that have come highest on the list, and let's see if we can get your answers. So first question is, did you look into the reasons why the regulated expression systems, the constructs, were particularly silenced by epigenetics?
Yeah, I mean, that's a great question. We looked at, a small number of things, but there are just so many reasons why there's epigenetic silence. So we actually tried. I mean, the reason this thing took so long is that we had basically, 1,000 different hypotheses about why we get epigenetic silencing, and we thought, oh, now we finally know what we're doing, and we actually tried it out, and then we failed and failed and failed and failed. So we thought it was because of bacterial sequences. We thought, GC islands. We thought, that we need to somehow maintain operin chromatin. We thought that if we have these pioneer factors, that be able to break through, and just nothing worked. I mean, and we haven't given up. It's not like we have given. We were very interested in epigenetic regulation, being able to overcome this. But we did discover that, kind of quote unquote a simpler approach, which is just to find promoters that are known to be constitutively active and able to resist epigenetic silencing, work extremely well, and they'll last forever. And so let's just try to solve the right problem. Yeah. But again, not giving up on epigenetics. I'm still, hopefully before I retire, we will have, amazing epigenetic control too. Yeah,
maybe Mo's group could sort something out for you.
Yeah.
Or
Right.
All right, next one. When do you think de novo engineered self-organization and pattern formation will make it into organoids and tissues?
March 15. No, I think soon. I think that there's a lot of activity right now that's just going to make that happen. So we work on a variety of different cell-cell communication mechanisms, like short distance, long distance. So we have things that diffuse over fairly long scales, and then there are people like Wendell Lim that are doing the SynNotch, so amazing systems. So, and he's been able to demonstrate some control. That Leonardo Morsat from his lab was also working and improving that. So spatial control is happening now. It is making its way into organoids. I would say that and Wendell, I think, showed actually some type of muscle differentiation circuit with spatial features. So I think it's going to slowly make its way into organoids, so we can actually reshape the way these organoids. So we're interested like in liver zonation and other kinds of specific patterns that we've already designed circuits to be able to do that. So it's going to happen. I would say on the order of a few years, certainly not decades.
Do you not have to fight the instinct of the cells to form their own structures and patterns that they're used to differentiating into?
Yeah, I mean that's a really important point, in that so we have kind of shifted the way we're thinking about it. So we don't want to fight with these systems. We want to collaborate with the cells, right? And so we have to figure out ways by which we can guide the cells. By way, synthetic biology is really not orthogonal, right? Synthetic biology is really, you have to really think about synthetic biology as being embedded into this complex environment. And so the question is, what are the mechanisms by which we can guide the system in particular ways? And there are ways that we can, I don't want to use the word fight, but we can have some pretty convincing arguments to the cells about what they should do. And we do this, and other people do this, that when you overexpress particular factors, you can really override a lot of signaling pathways. So there'll be some default behavior for the system, and you overexpress things. And interestingly enough, a lot of times you're able to actually, override what the cells would naturally do. So yes, I do believe that in many cases, we do have the ability to override what the cell would do naturally. Cool. It's just not easy.
We still got lots of questions coming in, but I'm going to skip to a small one who just wants something clarified, which I think is going to be a quick answer.
Okay.
What does DOX stand for in all of your slides? Yeah.
Doxycycline.
Oh, I knew that one. I got that as well. Doxycycline is it's an antibiotic, right? Like tetracycline antibiotic.
Yeah, it's a derivative of that. So doxycycline has been used for building the TET-on system. So it's actually, I think, the first that I know it's been used actually in the mid-90s or so. So it's actually, I think, one of the first molecules to be used to induce gene expression in mammalian systems. And it's just that, the TET-on, TET-off system, tried and true, amazing system, still incredibly powerful.
So out of all the advances mentioned today, which do you think could make a difference to patients first? First.
Okay, so now I have to guess what's going to get in first. So this is in terms of, so for example, the organoids, they're going to be very likely incorporated into drug development, fairly soon. So that has an indirect effect, I think, on patients. But I would imagine that the question was more about the direct effect, like, what kind of, so I think indirectly synthetic biology already has, some effect by allowing us to understand what's going on, improvements in drug development, use for biomanufacturing. So I think, that's happening now. That's beginning to take shape. I think that with respect to actual therapy, when are we going to see synthetic biology circuits make it into humans? My sense is that, for example, regulation over CAR T cells chimeric antigen receptor cells, that could be kind of the first big thing that where you have on switches, off switches, multi-input switches, safety switches. They're really going to harness the power and tame the power of CAR T cells.
I would be surprised actually if there wasn't a patient somewhere right now with a logic gate T cell.
Yeah,
experiment going on.
Yeah, so I think that some of these. So I'm not 100% sure, but I think some of those, simpler quote unquote CAR T logic circuits, they're either applying for phase 1 clinical trials or engaging in those. So that's really at the forefront.
Okay, next question. I think this is driven by a lot of our younger audience care less about healthcare and more about the environment.
Okay.
Is your kind of tissue organization being used in consumer biotech, for example, cultured meats for food?
Not yet, but I think it's directly. I think it's a great question, and actually something that we kind of were brainstorming. What else can we do with this? And literally just this past weekend, we said, oh, maybe we can use it for designer meats, designer hamburgers, because for them to really be to evolve properly, you need co-development in the cells and you need architecture. So that's a really great observation. Not being used yet, but I think something that could be applied quite soon.
Is that a bit too Californian for you guys over in Boston?
No, I don't know if I'm hip enough for that. That's a good question.
Okay, so someone asks on the phosphorylation circuit, is there excessive metabolic burden from this circuit in the yeast cells? Is that a concern and that limits the size of this kind of circuit?
Yeah, it's a great question. So basically the metabolic requirement here is mostly about the expression of the different enzymes. And the nice thing is that during the operation of the circuit, the metabolic load doesn't change that much, right? Because you have phosphorylation, dephosphorylation, but they don't seem to have, it's not like transcription and translation, which are really expensive. So the phosphorylation events that are quick and modify the state of the system don't require huge metabolic shift. So now the question then, with respect to the overall metabolic burden of the cell, on the cell, is how much of each protein do we need to produce in order to create robust circuitry? And with these phosphorylation mechanisms, you actually don't need very high levels of the individual proteins. Due to probably to the speed of the system and due to the kind of the noise attenuation properties. So you don't really need. We actually looked. We have a paper coming out on this stuff quite soon, hopefully within a few weeks. And we did look at the expression levels of the various genes that we're using, and they're really, we were able to use fairly weak promoters to be able to do that. So, an important question is, what's the dosage response of the system and the noise characteristics? And, they seem to be pretty good. But, I'm not going to come out and say that exactly what they are, but they anecdotally and empirically, they seem to not require significant expression levels.
Great. Do you have a few more minutes for a few more questions?
Yeah, I can hang out.
Great. So mammalian cells, so a few questions, particularly on the more complex circuits. What are the challenges for implementing these big complex circuits, particularly in mammalian cells compared to E. coli?
Yeah, great question. So a lot of challenges. So I guess, I don't know, do we have a few hours? It depends. One thing is delivery. This is actually not something we even talk about that much, but how do you get the DNA into the cells? Are you going to use transient transfection? Are you going to use lentivirus? Are you going to use piggyback? Are you going to use landing pad mechanisms? So even that initial decision of how to get the circuits into the cells is going to have a huge impact on the behavior of the cells, of the circuits. Finding elements that have good on-off responses is not necessarily that hard. You know, I spent a good amount of time on epigenetic silencing. What else? Orthogonality to the chassis is important. I would say that, interestingly, you know, and then also the speed, the fact that these, you know, the cells don't divide quite as often as bacterial cells, so you have to embed more kind of rapid mechanisms into the devices themselves. I would actually say, interestingly, there are a few things in mammalian synthetic biology that are easier than bacterial, and this is, you know, initially maybe a little counterintuitive, but for example, persistence of the circuits. So we can embed circuits into the genome, and we have circuits that are functional for many months and arguably even years. That's a little difficult to do in bacterial systems. So as long as these elements behave nicely with the cell, you know, they can persist for a long time. We don't have to worry about mutations that often. If it's in the genome, most likely it's going to be replicated, you know, with high fidelity in the genome for a very long time. Metabolic burden was another question that was asked. You know, we can reduce the expression levels of these sufficiently so that oftentimes the metabolic burden is not a huge burden. And so I think overall, it's very possible that the size of the circuits that we can build in mammalian systems can end up being much larger than what we can do with bacterial systems. So yeah, so there's a lot of you know, challenges, but I think there's some interesting opportunities there as well for creating different kinds of circuits. There's also lots of toys. So if you like genetic toys, man, this is you know, a nice sandbox, a nice toyland, or a nice toy store for you.
Well, the PERSIST toolbox looks amazing. That's a great addition as well. So a couple of questions now on the neural network stuff. Do you think the ANN demo will actually get extended into a viable computing system? In general, could you comment on computing with cells?
So I can almost guarantee that this will never replace computers. Right? I think, you know, when I started in synthetic biology, you know, people would occasionally say, oh yeah, you know, can do, you know, but you know, there's DNA computing or biological computing that would be faster. You know, yeah, I don't believe in that. You know, you know, so you really have to use what the substrate offers you. So DNA computing, you know, if you use it for the right reasons, it's amazing. I mean, you can do like DNA nanotechnology, or you can do DNA computing in contexts that you couldn't do before. So you have to think about, what is this really used for? So the artificial neuronal networks that we're building here are so slow, you know, that there's no way they're going to replace real computing, but they're also very powerful. So I think that as an abstraction for how to do sophisticated, powerful regulation in biological systems, I think that, you know, there's tremendous opportunities. There's tremendous, you know, number of situations whereby this kind of the notion of neuromorphic computing is much more suitable for doing like pattern classifiers, you know, response elements, and ultimately, you know, we want to be able to have circuits that learn and get better, right? So, you know, that's one of the hopes, is that, you know, we'll be able to do, you know, I showed very briefly DNA recombination, but, you know, there are a variety of other mechanisms, you know, including CRISPR and other kinds of mechanisms where we can control genetic mutations. And so imagine that we would be able to control, you know, have cells that can control their own genetic mutations, that they can actually evolve their circuitry over time and get better at it.
That would make my projects a lot easier.
Yeah.
Where do you think is the limit for the number of the microRNA inputs for a neural network?
It's large. I don't know. I would say that the limits are more on the internal neuronal network as opposed to the external. So if we view microRNAs as inputs, external inputs, then I think the internal topology structure network is going to be, kind of the limiting factor for some time. And then whatever size network we can build, we can put as many microRNA sensing mechanisms as we want. So right now, we can probably do with the existing devices that we have. I'm sure we can do 5, 6, 7 microRNA inputs, and then to get that number to be higher, would require expanding, for example, the endoribonuclease library. So I mentioned we have 9 right now. We're getting to about 15 soon. I think we'll be able to get with some effort, but I think we can get to 20, maybe 30, right? Not tomorrow. Not before the next time I visit the UK, but
it's still a pretty substantial number, I think.
Yeah,
yeah.
And so, yes, I think we can get there. These endoribonucleases are nice. They're just, they're easy to program. They're easy to engineer. You can measure orthogonality. And,
is there any off-target effects? So on mRNAs elsewhere in the human genome or transcriptome?
Yeah, some of them do it. You have to be very careful about that. So CSY4 in particular is an endoribonuclease that's used frequently, and that's actually the one we started with. And so if you use it at reasonable concentrations, it seems to have almost no effect or no measurable effect on the cell. But when you highly overexpress it, it seems to have, and at some point we actually looked at which specific endogenous RNAs it targets, but it does have an overall effect on the cell. And so CSY4 was our favorite, and we've actually, now our favorite is CASE, which at the same high levels of expression seems to have no observable effect on the endogenous pathways. And so, yes, you have to be, very careful. And as you incorporate new endoribonucleases, you really have to measure any kind of side effects they have.
All right, Ron, we're going to end with one last question, which is a bit more of a forward-thinking one. That's just popped up. To what extent do you think COVID and mRNA vaccines has opened a door for synthetic biology implementation in humans? Do you think we will see as much pushback as we have before in the future?
Maybe the anti-vaxxers will have a pushback, which is a huge population. It's a big problem in the U.S. right now. It's a huge problem in the U.S. But if you're, if you're a rational human being and not an anti-vaxxer, and I apologize if I offended anyone, but I'm really something that has been of huge concern to me and many other people in the U.S. right now, and it's a very, I think, very sad situation is what's going on. But I think for the people that accept, these life-saving mechanisms, there's a great acceptance of messenger RNA now. I mean, I wasn't even 100% sure about messenger RNA until I saw the data.
Yeah.
And I've been doing messenger RNA for a decade now. And RNA, so yes.
I mean, didn't your group have RNA plus protein circuits only? That you would show circuits working without delivering any DNA?
Oh yeah, for a decade. Yeah, for a decade. We have a startup that does this too. So
are they called Moderna? No.
Yeah, have you heard of that? Yeah. A little smaller.
Yeah, but it must be exciting to see that this can now be delivered.
It's amazing. I think that what Moderna and BioNTech have been able to demonstrate is unbelievable. And I mean it's transformative. And messenger RNA by a huge fraction of the population is now accepted as an intervention.
Yeah.
And that was not the case a year ago. And so is it going to be transformative to synthetic biology and the use of RNA? I hope so. I definitely anticipate it. So this notion of taking mRNA and being able to now do like microRNA sensing using mRNA, then you could get cell type specific expression. Yeah. Who's not going to want that, right?
Or yeah, or kind of conditional suicide of certain cell types, right?
Yeah, exactly.
Just by RNA delivery.
Yeah, exactly. So I think in terms of what is hopefully going to be, accepted, is not just the constitutive expression, but also some synthetic biology in there. And my guess is that, once there's the first real application that shows that, let's say microRNA classifier with RNA, people are gonna, and we're gonna be able to show, therapeutically that it helps, and it makes things safer, it makes things more effective. I think the barrier to acceptance is going to be relatively low. And so,
Great.
yeah. So
and probably barrier to investment as well, right?
Yeah, exactly. Yeah. So the one little silver lining in COVID, I guess. Yeah. So maybe in the long run, this will end up saving lives. So I don't know, but it's obviously a catastrophe, a world level catastrophe. But we try to do the best that we can, with the situation.
Okay, Ron, thanks for the wonderful talk and for staying for all these questions from all the students as well. And thank everyone for the great questions, all the SynBioUK and SynBIC students. And have a great afternoon, Ron, back over there in Boston, and all the best.
Thank you. Cheers, and enjoy the rest of the beer too.
Yeah, cheers.
I have my tea here, I guess.
Oh, I know you love tea, Ron.
Bye.
Bye.